Hey guys, it's Mr. Laka here to give you guys a little bit of read aloud, think aloud. We went over the PowerPoint today for the ruminant basic compartments, and so now what we're going to do is we're going to go a little more in depth and go over what the actual different ruminant species might need to sufficiently be maintaining their balance and overall composition. So let's go ahead and get started. This article is from Mississippi State University. I attached also a rubric for what we might end up doing this next week. So let's go ahead and get started. Ruminant feeding types is the name of the article. Based on the diets they prefer, ruminants can be classified into distinct feeding types: concentrate selectors, grass and forage eaters, and intermediate types. The relative sizes of various digestive system organs differ by ruminants' feeding type, creating differences in feeding adaptations. Knowledge of the grazing preferences and adaptations amongst ruminant livestock species helps in planning grazing systems for each individual species and also for multiple species grazing together or on the same acreage. So to put this into content, we have cows, goats, and sheep all grazing on the south pasture currently at the farm. So here's a little basic idea of what we're going to do. So concentrate selectors have a small reticulorumen in relation to their body size and selectively browse trees and shrubs. Deer and giraffes are examples of these concentrate selectors. Animals in this group of ruminant species select plants and plant parts high in accessible digestibility, nutrient-dense substances such as plant starch, protein, and fat. For example, deer prefer legumes over grasses. Concentrate selectors will very limited in their ability to digest the fibers and cellulose in plant cell walls. So my buddy Bucky up here, he likes to eat a lot of different trees and shrubs. In addition, he eats more clover from the pastures than what we find in these different other ones. So grass and roughage eaters include cattle and sheep. These ruminants depend on diets of grasses and other fibrous plant materials. They prefer diets such as fresh grasses over legumes, but can adequately manage rapidly fermenting feedstuffs. Grass and roughage eaters have a much longer intestine relative to their body length and a shorter proportion of large intestine to small intestine as compared to concentrate selectors. So the different organs on the inside of a deer compared to a cow, the cows are going to be a lot longer. In addition, they're built different body styles so they can contain all those different adequate parts. Goats are classified as intermediate types and prefer forbs and browse such as woody, shrubby type plants. This group of ruminants has adaptations of both concentrate selectors and grass roughage eaters. They have a fair, though limited, capacity to digest cellulose in plant cell walls. So like we did in the beginning of the year, we went over the idea of goats always eating garbage. Not always true. We want to make sure that we understand that goats can do both eat forage and shrubbery. They're kind of the cool breed out there. So let's go ahead and go into the carbohydrate digestion versus the protein digestion. Forages. On high forage diets, ruminants often ruminate or regurgitate ingested forage. This allows them to chew their cud to reduce the particle size and improve digestibility. As ruminants are transitioned to higher concentrate or a grain-based diet, they ruminate less. So when we take a cow and we take it from a grass-fed operation to a feedlot where they're fed a lot of corn, in addition they're fed a lot of different other things, but they have to take a little bit longer to adapt to the need of the forage. So going back to the reading, once inside the rectoreticulum, forage is exposed to a unique population of microbes that begin to ferment and digest the plant cell wall components and break these components down into acids to grow. The microbes ferment sugars to produce VFAs, acetate, propionate, and butyrate, methane, hydrogen sulfide, and carbon dioxide. The VFAs are then absorbed across the rumen wall, where then they go to the liver. Once at the liver, the VFAs are converted to glucose and glucogenesis. Because plant cell walls are slow to digest, this acid production is very slow. coupled with routine rumination, chewing, and rechewing of the cud that increases salivary flow, this makes for a rather stable pH environment around 6.0. So then we get into high concentration feedstuffs or grains. When ruminants are fed a high grain or concentrate rations, the digestion process is similar to forage digestion with few exceptions. Typically, on a high grain diet, there is less chewing and ruminating, which leads to less salivary production and buffering agents being produced. Additionally, most grains have a high concentration of readily digestible carbohydrates, unlike the more structural carbohydrates found in the plant cell walls. This readily digestible carbohydrate is rapidly digested, resulting in an increase of VFA production. The relative concentrations of VFAs are also changed, with propionate being produced in the greatest quantity, followed by acetate and butyrate. Less methane and heat are produced as well. The increase in VFA production leads to a more acidic environment, a pH of 5.5. It also causes a shift in microbial population by decreasing the forage using microbial population and potentially leading to a decrease in digestibility of forages. So if we have a cow on a high grain diet, it makes it a little bit tougher for them to digest the hay that we give to them in addition. Lactic acid, a strong acid, is a byproduct of starch fermentation. Lactic acid production, coupled with the increased VFA production, can overwhelm the ruminant's ability to buffer and absorb these acids and lead to a metabolic acidosis. The acidic environment leads to the tissue damage within the rumen and can lead to ulcerations of the rumen wall. Take care to provide adequate forage and avoid situations that might lead to acidosis when feeding ruminant high concentrate diets. Acidosis is discussed in detail in Mississippi State University Extension Service publication 2519, Beef Cattle Nutritional Disorders. In addition, energy as a nutrient in ruminant diets is discussed in detail in publication 2504, Energy in Beef Cattle Diets. We'll go into those probably a little bit later on in the semester, or probably next year when you guys take advanced animal science. So let's go ahead and go into the protein digestion. Two sources of protein are available for the ruminant to use: protein from the feed and microbial protein from the microbes that inhabit the rumen. A ruminant is unique in that its symbiotic relationship with these microbes. Like other living creatures, these microbes have requirements for protein and energy to facilitate growth and reproduction. During digestive tracts, some of these microorganisms are washed out of the rumen into the abomasum where they are digested like other proteins, thereby creating a source of protein for the animal. So remember that when we went over it today, we said that in the abomasum, that's where they create or obtain most of their protein from the feed, in addition to the rumen producing those microbes and bile. All crude protein, CP, the animal ingests is divided into two fractions: degradable intake protein, DIP, and undegradable intake protein, UIP, also called rumen bypass protein. Each feedstuff, such as cottonseed meal, soybean hulls, and annual ryegrass forage, has different proportions of each protein type. Rumen microbes break down the DIP into ammonia, amino acids, and peptides, which are used by the microbes along with the energy from carbohydrate digestion for growth and reproduction. Excess ammonia is absorbed through the rumen wall and converted into urea in the liver, where it returns to the blood, to the saliva, or is secreted by the body. Urea toxicity comes from overfeeding urea to ruminants. Ingested urea is immediately degraded to ammonia in the rumen. So when we want to make sure that we don't get a problem with urea, we give them a little shot of ammonia and they can absorb it a little bit better. So when ammonia, then energy is available for building protein from the nitrogen supplied by urea, the excess ammonia is absorbed through the rumen wall. Toxicity occurs when the excess ammonia overwhelms the liver's ability to detoxify it into urea. This can kill the animal, however, With sufficient energy, microbes use ammonia and amino acids to grow and reproduce. The rumen does not degrade the UIP component of feedstuffs. The UIP bypasses the rumen and makes its way into the omasum to the abomasum. In the abomasum, the ruminant uses UIP along with the microorganisms washed out of the rumen as a protein source. Protein as a nutrient in ruminant diets is discussed in detail in the Mississippi State University Extension Service publication 2499, Protein in Beef Cattle Diets. So I hope this helps you guys understand the differences between cattle, goats, and deer as a ruminant species. So when we come to class tomorrow, I hope you guys all take a look at this, and we can go from there. Thanks, guys.